Good day, this is Professor Will, CBMD PhD. Today is Monday, October 19, 2020. It is 6:43 and 51 seconds p.m. Eastern Standard Time. And this is a continuation of my microbiological research, so let's hop to it. We may define systematics as a scientific study of organisms, with the ultimate object of characterizing and arranging them in an orderly manner. Systematics also might be defined as the study of organismal diversity and interrelationships. Systematics includes taxonomy with its subdivisions, and includes aspects of ecology, biochemistry, genetics, pathology, molecular biology, and microscopy. Taxonomy is often used as a synonym for systematics. Taxonomy as the art of biological classification, as a theoretical study of classification includes its basis, principles, and rules. 1. Classification, which means the orderly arrangement of units into groups. 2. Nomenclature, which means the labeling of units defined by classification. 3. Identification of unknowns with the units defined by classification and labeled by nomenclature, i.e. identification is the practical application of the arts of classification and nomenclature. Classification is often confused with identification, because identification may be understood either transitively, identification of unknowns with units defined by classification, or intransitively, to describe the identity of a species as such and use it as a basis for classification. Consider the two functions of taxonomy to be: to identify and describe the basic taxonomical units or species, and 2. to devise an appropriate way of arranging and cataloging these units. Rules about the rank of taxa, naming of taxa, nomenclature, types and their designation, priority and publication of names, citation of authors and names, changes in names of taxa as a result of transference, union of change in rank, rules about illegitimate names, and epithets, replacement, rejection, and conservation of names and epithets. Also a number of rules pertain to the correct usage of Latin orthography. The rules of International Code of Nomenclature Bacteria are designed to make effective the principles. They are internationally accepted and must be obeyed in bacterial nomenclature, even retrospectively. Bacterial nomenclature is not completely independent for practical reasons. Bacteria may not bear names that are validly used for algae, fungi, protozoa, or viruses. In the code, the word taxon, plural taxa, is used for any taxonomic group of organisms. A taxon consists of one or more elements. For each named taxon of various taxonomic categories, there shall be designated a nomenclature type, that is, the element of the taxon to which the name is permanently associated. The taxonomic categories form a hierarchy. In general, little use is made of subcategories such as suborder and subfamily, or of the category tribe. The basis of the taxonomic hierarchy is essentially the species. In 1968, researchers humorously summarized a species as a group of organisms defined more or less subjectively by the criteria chosen by the taxonomists to show to best advantage as far as possible in putting into practice their individual concept of what a species is. A species consists of an assemblage of individuals, or in microorganisms of clonal populations that share a high degree of phenotypic similarity, coupled with an appreciable dissimilarity from other assemblages of the same general kind. The modern phylogenetic definition of species is based on DNA hybridization procedures and generally would include all strains with approximately 70% or greater DNA relatedness with 5 degrees Celsius or less delta Tm. Both values must be considered. Phenotypic characteristics should agree with this definition and would be allowed to override the phylogenetic concept of species only in a few exceptional cases. It is recommended that a distinct genospecies, i.e. a species discernible only by nucleic acid comparisons, that cannot be differentiated from another genospecies on the basis of any known phenotypic property, not be named until some phenotypic differentiating property is found. When a new species is described, the type must be indicated. Whenever possible, the type should be a pure culture designated as a type strain. For a species described from impure culture, a description as preserved specimen or illustration may serve as a type rule 18a. If such an organism is brought into pure culture later on, it may be designated as a type strain and replace the original type rule 18h. Before publication of the name of a new species, a culture of the type strain should be deposited in at least one of the permanently established culture collections from which it is readily available. The strain number given by the culture collection should be quoted in the publication description recommendation 38. Good day, this is Professor Will, CBMD PhD on microbiology, bioinformatics, and computer science. I hope you enjoyed listening. Until next time, have a great day.